All right. Hi, I'm David Luecke. I'm talking about changes in growth that have been produced by the evolution of relative organ size. All right, so as evolutionary biologists, we're very interested in explaining how diversity has the origins of diversity across life. This figure, something like it, is in basically every evolution textbook, and it depicts the pervasive homology that is underpinning the vast disparate structures. But it also shows us the power that we have in changing the relative sizes of body parts to produce different morphologies and functions. So I study this phenomenon in the species Drosophila prolongata. So males of the species have evolved radically larger first legs in the last few million years since they've diverged from their sister lineage here depicted by the sister group unnamed species KB866. So to consider how evolution has modified development to produce this shift, we need to think about how development is controlling organ size. So organs have an intrinsic size to which they will grow and stop growing without any external signals. They also integrate an extrinsic cue that scales with body size to enable them the final size of the organ to scale with total body size. So evolution can modify either of these two components, but we can distinguish between these possibilities by looking at population phenotypic data. So here I have shown the length of male first leg from 6 different Drosophila species plotted against their thorax lengths. So we see that there's broad conservation in the relative size of the first legs and in the scaling relationship. So if evolution were modifying relative organ size by affecting that extrinsic body size signal, then we would expect larger individuals to gain disproportionately. And the slope of the scaling relationship would increase considerably. So this is what's happened in horned beetles, who have evolved an increased sensitivity to insulin signaling. If, on the other hand, the intrinsic organ size has evolved, we would expect all individuals, regardless of their body size, to gain to the same degree. So in Drosophila prolongata, we see that there is no real change in the scaling of the slope. Everybody has gained proportionally, regardless of their body size, indicating that this is evolution of that intrinsic size of the first leg organ. So how has development produced this change? Well, we know that since first legs end up larger than second legs, the growth rate of first legs must at some point be higher than the growth rate of second legs. We don't know if this is consistent across development or if there is a temporally constrained growth spurt. So in whole metabolous insects, we grow our adult structures in the larva in the form of these imaginal discs. So the pre-grown tissues during metamorphosis rearrange to form the legs, the eyes, the wings, the genitals, etc. So I dissected out the first and second leg imaginal discs from across the developmental time course of the final larval instar and stained them with markers for all cells and with a marker for mitotic cells. So this enables measurements of the organ size, the number of nuclei, a proxy of the instantaneous proliferation rate, the number of mitotic cells, and then the mitotic index, which is the proportion of cells that are undergoing division. So if we take that number of dividing cells as a proxy for the growth rate, subtract the number of dividing cells in the second leg from the number of dividing cells in the first leg, we get a proxy for the first leg bias of growth. So in KB866, we see that there is basically no bias to growth in the first leg or the second leg. In Drosophila prolongata, we see a very significant increase, first leg bias growth, and this persists throughout development. Oh, God. So this enables us to grow larger first legs gradually over the course of development. So how is growth rate regulated? Well, it's a complicated process. One very interesting aspect is the coordination of developing organs. If one organ gets too far ahead or behind, it will compensate its growth. And we see this signal in our disc data. It's a negative correlation between the ratio between organ size and the ratio of the mitotic indices. So where this this is in KB866, where this negative correlation crosses the isogrowth line, the x-axis, is the end dynamics of these growth, the end point of these growth dynamics. So prolongata has this same negative correlation, but the point has shifted to the right, indicating that the long-term dynamics have also shifted. So this is consistent with a change in the intrinsic growth rate that is not just about the final size, but throughout development, the relative sizes of these developing organs. So this is an absolutely prolific way, and this is a good system, and thanks everybody.